Good evening. Thank you for joining this session. Today, I would like to talk you about a topic which is usually second uh, big biology-related topic what data analysts encounter. Today, we will talk about not the sequence aspects of genes and proteins. Instead, we will focus on the annotation. Annotation is, um, as we have discussed last week, is very important to record as the related information and knowledge related to the sequences what we handle in many data analysis scenarios. Now, if you are a bit unsure that what we are doing here today, think about things like if you identify a big group of genes or proteins which are relevant or interesting for your study or research or development project, then obviously you want to somehow find that what are the common amongst them. For example, imagine an RNA-seq experiment where you identify a group of genes which are responding strongly to a drug treatment, or you are able to identify a group of proteins which are highly expressed in a certain disease. You can imagine things like that. Then the next step is to try to find some kind of common aspects, common features of those genes. So we need the annotation for each or most of the genes involved in the study to find that what is common among them. So just imagine that in old days research, what we have studied all in high school, like Mendel's experiments with the peas and the Drosophila experiments and so on. Then in those old days, genetics related research, we operated with a maximum of a handful of genes. So you remember a single locus, two or maximum three loci in case of Mendel experiments or the Drosophila experiments. It was relatively easy to see that what these genes are doing. So if you mutate that gene, that the Drosophila will have white eye instead of red eye, or curly wing, and that kind of simple assignment. So it was sort of easy to understand that this gene has some kind of function related to this phenotype. Or if you remember the lacoperon from the Escherichia coli, you remember that if you mutate the LAC gene, then the Escherichia coli is not able to grow anymore on lactose sugar. So it's easy to understand that, okay, those genes are related somehow to lactose digestion in the bacteria. So nowadays, we are not, the present day research situations are not so easy. When you perform a typical high throughput experiment, you can end up very easily 100, 200, 400 significantly differentially expressed genes. Or if you think about genome analysis, you can very easily end up with a large list of genes which is related to some kind of disease phenotype, if we are talking about this kind of genome-wise association studies. So we will use, or I will show you today, how to use annotation and analysis of this annotation of these genes to identify such features which are enriched among those genes what we select from a larger background population. Very often today we identify a handful of genes from the entire genome. So enrichment analysis, as you see on the screen, is basically nothing else than helping us to interpret a list of genes. We try to find common features of those. And when we are doing this in practice, we use a particular kind of annotation, for example, gene ontologies or pathways, for example, KEC pathways, transcription factor binding sites, microRNAs. And among experimental biology researchers, enrichment analysis is labeled like ontology analysis or pathway analysis. Probably you have heard these terms instead of the more general enrichment analysis, which describes all of these. But the basic research question, what we ask in all of these analysis scenarios, that what is the distinguishing feature associated strongly to our target gene list. So I would like today to introduce you two different approaches, two methods. Both of them are rather popular nowadays. So both of them are in active use. One of them is called the overrepresentation analysis, and the other one is called gene set enrichment analysis. They are working in slightly different ways, and the most important is that what kind of input should we provide to these algorithms. When we are performing overrepresentation analysis, we have to compare a target gene list, which is the list of genes of our interest, to some kind of background list. Very often this background list is the entire genome, but not always. Sometimes it makes sense to identify a smaller set of genes as a background target. I will show you examples for that. Okay, so target gene list and background gene list, and we try to compare the target annotation terms associated with the target genes to the entire background set. On the contrary, gene set enrichment analysis, it is working a slightly different way. You can score the importance of a single annotation term, but for that you need a list of ranked genes. And you get this kind of ranked genes, for example, from any kind of differential gene expression analysis, because you can rank the genes either using the adjusted p-value or using the differential expression ratio, so the fold chain, how many times the genes upregulated or downregulated. You have a list of genes and you just order based on that. On the top of the list you have the most upregulated genes, and on the bottom of the list you have the most downregulated genes. So these are two rather important approach differences, and I would like to show you how these are working in practice. But before I go to there, I would like to highlight a bit some of the annotations, some of the features of the annotations, and I will use the gene ontologies as an example annotation, because, well, that is one of the most often used kind of annotation what we use. Now, I really hope that you remember from Marty's lectures the most important features, what gene ontologies are. Do you? This is a question. Do you remember what gene ontologies are and how they are used?
Yes.
Okay, very good. So I really don't want to go into the details. I will just highlight the most important features what we will use here. So the important ideas here that there are different kind of gene ontologies like cellular components, molecular functions, biological processes, and the individual gene ontology terms, they have relationships to each other. And how I sort of interpret this, that gene ontologies in general, they have a structure. So we have to take into account this kind of structure when we are working with GOs. So this is a very, very small part of the general gene ontology terms. So you can see that, for example, these are some of the biological processes. This is actually a figure from one of my research studies. So you can see that biological processes, there are subterms like immune system processes, regulation of biological processes, response to stimuli. And then some of the terms or subterms have even sub-subterms under them. What is important that terms at higher level and terms at lower level are different, usually in that they terms at lower level, they are more specific. They annotate fewer genes, like obviously biological regulation is a very, very wide term, while regulation of biological processes is more narrow. Also, if you have a look at the immune response term, obviously there are some genes then, but a regulation of immune responses, which is under, it's a more narrow category. Now, this structure is very specific. So it's not just a tree. You can see that there are certain terms at lower level which can belong to multiple higher level categories, and obviously a higher level term can have multiple lower level terms. But it is important that these relationships has a direction. So for example, a lower level category belongs under, is part of a higher level category. Okay, so it means that although unfortunately on this figure it's not shown, but these connections are actually arrows. So you can start from the highest level and you can always walk along or into the directions of these arrows, and you can reach more specific terms. But it is very important to understand that there are no circles. So you cannot start from here and walk around and reach back. So it never happens. So that is very specific to the structure of gene ontology terms, or these subtrees or substructures of gene ontologies. And the name for this is these directed acyclic graphs. So directed because there is a direction incorporated in the relationships, but acyclic meaning that you are not able to walk back to your original starting point along using only the directions of the arrows. So What we want to do is to try to, when we perform enrichment analysis using gene ontologies, what we want to do to find terms which are furthest, possibly furthest away from the highest term. So the most specific terms which are significantly associated with your target gene list. And this is sort of obvious, so you are not really, it's not very helpful to find a list of genes which are associated with biological regulation. So that's really not meaningful to find that kind of description of your target genes. But if you find something that, hey, there are 14 genes among my gene list which are associated with inflammatory response, now that is something. Okay, so you can figure that, hey, this my experiments has something to do with inflammatory response or IgG binding. So obviously this is an immunology study what we have performed here. So let's have a look at the workflow, general workflow of enrichment analysis. First you calculate some kind of enrichment score, for example, overrepresentation or association in the two methods what we will cover. Then you, independently from the enrichment calculation, you perform a redundancy filtering. For example, what I have just explained, in case of gene ontology, you take into account the structure of this directed acyclic graph, and you try to climb down on the ontology structure to find the most specific terms which are still significantly associated with your gene list. And when we are doing this, we perform lots of hypothesis testing calculations. And how we have discussed in the statistics course, that's always arises the risk of having false, positive, negative results. That's why you have to always perform a p-value adjustment in this case. Okay, so the three steps: some kind of enrichment calculation, redundancy filtering, and adjusting the p-values, taking into account the number of multiple hypotheses that you have just performed. Okay, let's start with the specifics of overrepresentation analysis. So I will walk you through how to do the actual calculations, because at the end of the day they are relatively simple, and then I will show you a bit of demo how to do it with R for real. So overrepresentation analysis, just again, you need a target gene, a list of target genes, the genes, for example, what you have identified from your RNA-seq or microarray experiments, that, hey, these are the genes which are behaving differently if I treat them or if they are from disease patients and so on. Then you can have a reference gene list. For the sake of simplicity, we can use the entire genome. You need to have the total number of annotated genes. So we consider one annotation term at a time. This is an iterative process. So if you remember, or if we go back to this, these all rectangles are representing ontology terms. So you do these calculations, all of these terms one by one. So you need to know that how many, if you consider one annotation, how many genes in the entire list, reference list, is annotated with them. And using these numbers, you can calculate the expected number of annotated genes on your target list. Okay, just one small note that I have these terms probe here on the slide because we can perform this also if you are using microarray experiments to obtain your gene list. You do not have to convert probes to genes, if you remember, and we will cover this in the second course, the high throughput course, that microarrays are using probes. So this works exactly the same way, regardless if you use genes, probes, exons, individual exons even. So it transcripts, it doesn't really matter. We are talking about genes for the sake of simplicity. Okay, so let's have a look on at an example. I'm interested in the annotation term of glutathione metabolism. So I interested that so I have a list of genes from an experiment which contains 120 genes, and I'm interested, or I'm testing the idea if they are associated with this glutathione metabolism annotation term. And this is actually coming from the KEGG database. And these are human genes in this experiment. So the KEGG database holds information about 20,124 human genes at the time when I have prepared this calculation. And 39 of those annotated with this term glutathione metabolism genes. So I have my own target genes, which is 120, and 30 of those are annotated with glutathione metabolism. So just have a look intuitively. We have 20,000, around 20,000 genes in human genome. I have picked up 120 of those using some kind of experiment. And I know that in the entire genome, there are approximately 40 glutathione metabolism genes. And my experiment picked 30 out of those 39. So intuitively, I would say that, hey, this means something. So I was able to pinpoint with my experiment 30 out of 39 glutathione metabolism genes. So I'm arguing that that my experiment affects glutathione metabolism. But can I prove it numerically using statistics? So let's see. So the expected number of genes is pretty easy to calculate. So just calculate that what is the fraction of the entire human genome, 20,000 human genes in the human genome, which are annotated with glutathione metabolism. That's the probability. This fraction is the probability that a randomly pinpointed gene is actually glutathione metabolism gene. You just multiply that with the size of your target gene lists. There are 120 genes on your list. So this simple calculation will give you that the probability, the expected number of genes of a randomly selected 120 genes from this human genome background contains approximately 1/4, so less than one glutathione metabolism gene. So our intuition was well placed. If we are expected to to see 0.23, so less than one gene on a list of 120 target genes. And instead of that, we are observing 30, then there is a high probability that this is something significant. We can calculate the so-called enrichment level, which is just telling us that how many times more we observe than we have expected. So we just basically calculated the fraction of actually observed number of genes and the expected number of genes. And we can tell that, hey, we see 129 times more than we have expected. So this is a pretty convincing enrichment level. So we see in this target gene list 129 times more glutathione metabolism genes that what we would see if it would just randomly pick up 120 genes. But the question mark remains, is it significant? So to answer that kind of questions, we have to use a statistical test to provide the probability that what is the probability that even though we observe 129 times enrichment, it is just because of the game of probability, just because we were very lucky. We picked up those. You know, some people even win the lottery, so it's not impossible that you just treat your samples with distilled water and still you will observe this kind of enrichment. With statistical tests, we can calculate the probability that this would happen. And the tool for that is called the hypergeometric test. Hypergeometric test is a general tool. It's not biology specific, not enrichment specific. It is basically it serves a purpose that for a given target gene list, we can ask that how probable that we would get such a list with such an enrichment ratio just by pure luck. And for that, we can have this probability of successes, the number of draws without replacement, and so on and so on. So the hypergeometric distribution itself is giving the distribution of the number of successes if you draw more and more and more genes. For example, if you want to pinpoint a single gene, then you have to, if you draw like half of the genome, 10,000 genes, then you will have 50% probability that you will pick up that particular. And you can generalize this, that if you have 39 genes and you want to obtain 30 of those, how many you have to draw that to get those exactly that numbers with a certain probability. And when you have exactly the numbers that, okay, you have drawn 120 and you observe 30 of those, the hypergeometric test will tell you that what is the raw probability that it will happen just by chance. Okay, so in R, because hypergeometric test is just a general statistical test, is available as a general statistical test in R, the P hyper, and you have to provide exactly these parameters, which is Q is the vector of quantities representing the number of successes. So first, this is 30, which is the number of success, meaning that we were able to identify this many number of genes successfully out of 39. And we can tell that actually there are 39 such genes and 20,124 minus 39 non-glutathione related genes, and that we have drawn 120 genes. What you see here is that the probability that this has happened just by chance is very, very low. You read this, that 9.4 times 10 on the power of minus 64. So this is a crazily good p-value. So the probability that it has happened just by chance is really less than the second in the age of the universe or something like that. So very low probability. So what we see here is that we were able to calculate that the enrichment ratio is 129 times for this particular annotation term in the case of this gene list, and this result is significant with this particular p-value. But, and this is a big issue, we still have to take into account redundancy filtering and the adjusting the p-values. So in practice, when we are doing this, we have a long list of annotation terms. Sometimes the entire all the terms on the ontology list or list of gene ontologies, meaning hundreds and thousands of gene expression terms. And we will perform these calculations for all of those. And we will calculate the p-value for all of those annotation terms, meaning that we have performed lots of hypothesis testings. So that's why it is prudent and important that we adjust the p-value as we have discussed on the statistics course. So in this slide, this is again the same structure. We can see how to take into account the structure, this cyclic acyclic graph nature of gene ontologies. So you see in completely analog analysis that in an analysis I have identified 26 genes which were associated with immune system process. 21 of those were in immune response, 10 of those were in regulation of immune response, and 9 of those were under the label of positive regulation of immune response. So you see that how I try to go for the most specific term, which is still significant. If you observe, the adjusted p-values are breaking down. So the 26 genes related to immune system process is having a very, very good p-value. But if I'm going down in the structure, the p-value is actually increasing. So this power number is going closer to 0. So you see that for the 9 positive regulation of immune response genes, the p-value already in the 10 on the power of minus 8 level. So of course that is still significant. That's why this is highlighted with red. On the contrary, the black terms are they lost their significant status. So they are not significant, even though they are under and more specific than the terms above them. So exactly that's how you try to take into account the most specific terms and try to find those genes which are relevant there. Okay, there are different kind of algorithms which are able to do that. So we do not do this manually, because that would be a crazy amount of comparisons among the higher and lower level terms. So we have in R and in general, we have algorithms which are able to narrow down the scope of the ontology terms in this process. Yeah, and the last step is adjusting for the multiple hypothesis testing. Uh, just very briefly, let's repeat why what are the important things to know about them. So the individual hypothesis tests, in this case the individual hypergeometric tests, they are giving you a raw p-value which is true and valid for that individual test. But if you perform too many, in principle more than 10, 20 hypothesis tests in parallel, the the probability of making an error and including terms incorrectly to your result list is adding together. And if you use a p-value threshold 5%, then only after 20 such tests it adds up to 1. So you can be sure that at least one of the terms what you have identified that way will be wrong. So that's why we never use the raw p-values from this kind of calculations. Instead, we use the so-called adjusted p-values. Now, there are so many different methods to adjust p-values in general, that it is important to understand that in biology we use only one of the two options. Either we use the so-called Bonferroni method, which is very strict. Basically, it is excluding, so decreasing the threshold for the significance by multiplying threshold with the number of tests, actually dividing. But anyway, the point here is that if you have to use, or you should use the Bonferroni method if life or death depends on your results. So in practice, it is only used in kind of drug test kind of analysis, where a patient would die if the result would be wrong. So in practice, so the trade-off with the Bonferroni method, that especially because it is strict, it will throw away a lot of otherwise correct conclusion. So you can accept that it is very sure if an adjusted p-value is still significant when you use the Bonferroni method, but unfortunately it means that you will throw away lots of good results. So the method what is usually used to address this is the so-called Benjamini-Hochberg method, or also known as the false discovery rate, which is not an entirely correct term. False discovery rate is used to calculate the adjusted p-value, but it's not the same thing. But anyway, very often you just see that instead of adjusted p-value, you see that FDR, false discovery rate. So this is the most popular solution to this problem. And when you perform research, and it is not that important that if you make some mistakes in your calculations, so some of your results will be incorrect, that this is the one I recommend you to use. Yeah, you can use the p-adjust function in R to address this issue, and and I will show you. No, I won't. I have shown that. Okay, so I will show you a demo exactly how to do overrepresentation analysis using R. So I will share my screen. Studio, yes. So yeah, it's visible. Okay, so here I will use human gene set to uh, to do this analysis, and I will show you what is here. Basically, this is just a list of interest gene IDs. You remember from Marty's course, interest gene IDs are identifying actual genes. And I want to find, so this is a list of genes which I have used in one of my previous research projects. We call this the immunome. So it contains approximately 900 human genes which are associated with immune system functions. So I would like to see what kind of annotation terms are associated with this immunome genes. So just again, 900 genes out of the 20,000 entire human gene set. So I will use a database file which is installed as an R package, org.hs.eg.db, and I'm just picking up a list of annotation. Here you can see that there are genes, gene symbols, interest gene IDs, and individual gene ontology IDs here. Approximately that's what we can observe there. So we can do the same, for example, to pathways. So this is basically the same idea. Chromosomal locations. Chromosomal locations again. These are the chromosome numbers. These are the gene names. These are the positions of starting of that gene. So we have the known information about those, and we would like to associate that with the immunome genes, and we will use the TopGo package, which is and one of the implementations of the overrepresentation analysis. And the exact procedure is not very important. I just want to show you what kind of results we will get. So remember we have a target list and a background list, and here is what we get after performing the overrepresentation analysis. We have a table where the individual rows are on ontology terms. For example, this one is this GOID. This is the toll-like receptor binding ontology. And on our background list, there are 9 such genes, and there are 9 such genes on our target list. So you can see that we were able to pinpoint all of those. So if we calculate the size of the target gene list and the background gene list, we have expected to see only 1.7. Instead of that, we have seen 9. So we can calculate the enrichment ratio and the p-value here. So you can see that this is significant. Not very surprising that the toll-like receptor binding is important in immune functions in this particular analysis. Okay, so what I just wanted to show you that basically at the end of overrepresentation analysis, you get a ranked list of gene ontology terms or other annotation terms. All right, let's go back to the other method. So we have had an overview of overrepresentation analysis. Now let's have a look at gene set enrichment analysis. So the issue with overrepresentation analysis is that it handles the target genes uniformly. So even if you have some kind of value which is differentiate among those genes, for example, some of those genes are showing a very, very high differential gene expression, so log FC fold chain ratio, so upregulated by 20 times, and the other gene is just upregulated a little bit. Overrepresentation analysis, it doesn't care about that. It handles those equally. So this is the issue what gene set enrichment analysis tries to address. It tries to see if there is some kind of typical distribution along a list of ranked genes or probes. So these kind of gene lists are just again very typical from high throughput experiments, because very often you get this information that what is the upregulation, downregulation level, how big the differential gene expression, and you just order those genes using that information. And just again, we will calculate perform a calculation for each annotation terms one by one. And we will compare that calculation or the results of that calculation to random gene lists. So when we randomize the order of that gene list, and we want to see if this score what we have calculated is similar or not. Of course, because we perform this for many terms multiple times, we have to adjust the p-values. Okay, so let's see in practice. We have a list of genes. They are annotated by gene ID here again, and some of those genes are MAP kinase related genes, and you put these labels in the second column of a table. So the order is important here. For example, if this order is representing differential gene expression, it is customary that the gene on the top is the one where we observed the highest upregulation, and the gene on the bottom is the one where we observed the highest downregulation. Okay, so basically the gene expression difference here that on the top very much going up, and the genes on the bottom, they are going down. Okay, so what we do is just basically pick up this order. We pick up this order and we just put it on the x-axis of this diagram. So what was our top gene? Here, it will come here. So how we perform this calculation is that we set so-called running enrichment score, what we are visualizing on the y-axis, and we set it to 0 for the first gene, and we start to step down on this list one by one. And when we encounter a gene where we observe that this label is labeling that gene, then we are going up. And when we observe a gene where this is missing, okay, I will make it a different color. So this is missing, then we go down. So this is basically so-called, that's why it's called a running enrichment score, because you are calculating always taking into account the previous value. So you start from top, stepping down, and when you see MapK pathway, you are drawing the line up, and when you do not see MapK pathway, you are drawing the line down. So this is the actual calculation method, and at the end of the day, you get this kind of some kind of shape here, and you can get what you want to calculate is that what was the maximum of the enrichment score when it was furthest away from the x-axis. So it can happen in two different situations. Either those terms are associated with the first part, so the top of your list, then you will observe this big hill. Then the running enrichment score will have a maximum, because that's the top of the hill will be furthest away from the x-axis. Another option, then you will find only this kind of annotated genes at the end of your gene list, meaning the bottom of your original list. Then first your running enrichment score will go to minus, and then it will climb back to 0. In this case, your enrichment score will have a minimum, because it will be basically a valley instead of a peak. So at the bottom of the valley will be the situation where the enrichment score is furthest away from the 0 axis. Okay, let's have a look at what do these things mean. So obviously, I will highlight a bit. So this was the MAP kinase signaling pathway example. So this is the same graph, but perhaps we have find a similar one. Yeah. So this is the same set. Cytokine receptor interaction. So this case, it looks very similar. So most of the annotated genes are on the top, meaning that these annotation term or genes related to these annotation terms were upregulated or very highly upregulated, mostly in your experiment. So that's why you can see a definite peak in the positive region. On the contrary, when most of the genes are associated with the mostly downregulated genes, which is the bottom of the list or right hand of the x-axis, you will have a very, very deep valley, and the enrichment score will be negative. Just like in the case of this Huntington's disease or oxidative phosphorylation. Okay, so maybe this juxtat signaling pathway is even better example, because this was selected so that the MAP kinase signaling pathway is upregulated. So from such an experiment, this data is coming from. Obviously, the juxtat signaling pathway is full of MAP kinases. So when those genes are upregulated, obviously that the MAP kinase signaling pathway related genes are upregulated too, because basically they are same or almost the same. Okay, so basically what you can do is just you will get such a graph for each and individual term what you have tested. So basically just by a visual inspection you can identify this kind of cases when some term is associated only with the upregulated or mostly with the downregulated genes. And you can use simulations like picking up or randomizing the list or picking up random lists to figure out if these enrichment scores, which is at the end result of this calculation, is significant or not. So you are able to associate a p-value also with these individual plots. Then you can filter out those which are not significant. So you can see only those annotation terms where you have a definite peak or valley. Because if an annotation term is not particularly associated to any part of your plot, then it will just be a noise around zero. It will go up and down, and you won't get a definite enrichment score, and the p-value will be bad. You will be able to filter it out. Okay, at the end of this exercise, it doesn't matter if you have used overrepresentation analysis or enrichment analysis. You will have a list of annotation terms which are associated to your gene list. It is very important not just to mechanically accept the results of this kind of analysis. You have to use always common sense to interpret the results and to evaluate if those annotation terms, which are really highly enriched or associated with your genes, if they are meaningful for the experiments, if they carry forward your work or not. You have to consider that mathematical and statistical significance, it doesn't mean biological relevance. I cannot stop to stress this issue. For example, if you, on the slide, if you study gout patients and you find genes which are upregulated, downregulated, or differentially expressed in gout patients with active symptoms, and you perform enrichment analysis, and you get an enriched term purine metabolism, then what you have figured out, everyone knows that gout is a purine metabolism disease. So yeah, the result is statistically significant, but who cares? So you have to always take time and sit down to understand if that result carries forward your research or your study or not. The same way, it is possible to have results something like in the second example, that you can get the notch signaling pathway enriched among your genes. There are 48 such genes in the human genome. So if you analyze only a small target gene set, it can happen that you will find 4 of those 48 genes on your list. Can you say honestly that 4 out of 48 genes signifies biological relevance? So you have found like 46 other genes in your experiment. So most probably, yeah, it's somehow involved, but not very much. Okay, so you have to think very carefully how to interpret the results of the enrichment analysis. All right, that was the part I wanted to tell you about annotation, how to analyze annotation. Again, in the Moodle material, you will see examples how to obtain annotation data for a set of genes, for a set of proteins, how to perform both the overrepresentation analysis or the gene set enrichment analysis for real data sets. You will be able to practice that.